All right, so next up is Anne O'Donnell Luria, who is our Associate Director here at the Center for Mendelian Genomics. And she will be talking about annotation data and its uses, which I realize I just basically read your intro slide, but here is Anne.
Thanks. Hi. Okay, so I talk about using GENOMES a lot, but I'm going to talk about all of the different annotation data sets that we use when we analyze our data, and my hope is to prepare everyone for the workshop that comes after this. So I'm going to talk, I can't cover all of this obviously, so I'm going to talk about annotation data and how we use it, introduce the concept of transcripts, genomic reference databases, in silico prediction and conservation, expression databases, which you've heard some about, GTEx and RNA-seq, and variant interpretation databases, which you'll be hearing a lot more about later. My goal with all of this is to kind of introduce you to the concept and then think a little bit about how we use it or think about them and then move on to the next concept. So let's go through it. So in general, all of the data we get from exome or genome sequencing goes into a variant call file, which you've heard about already, and it lists all the places that the genomic sequence in your patient differs from the reference sequence. So it's very structured information, and you can annotate your VCF in a number of ways. You can describe the type of variant you're seeing, what transcripts the variant falls in, the allele frequencies of the variant in the general population, and in silico predictions along with many other features. So there's a few things about variant nomenclature that are really critical so that we're all comparing the same variants when we have our VCFs. So one of the things is in clinical interpretation, we tend to use HGVS as the standard nomenclature. And this comes up as a point of confusion when we talk about things like frame shifts or other types of variants that are indels, because the HGVS likes to align things to the right and a VCF likes to align things to the left. And it doesn't seem like a big deal. People are left right-handed. Why does it matter? And a classic example is the CFTR gene here. So it's a phenylalanine 508 deletion. It's an in-frame deletion. And yet HGVS nomenclature gives it totally different genomic coordinates than a VCF does. And in a sort of non-intuitive way in that it's not even like one base pair off, it's two base pairs off, which even as I was putting this together, I was like, wait, I had to stop and like actually phone a friend to get some help with how this works again, because it is very non-intuitive. And so what's happening here is what you're seeing is once you delete your 3 base pairs, you're left with 2 T's, but exactly which T's are left, who's to say? No one really knows. And so a VCF assumes that the leftmost 3 bases that can be deleted are gone, and that we're left with these T's. Well, HGVS assumes these 3 over here. And so this just highlights how important it is that we're all calling variants and describing variants in the same way, or you'd be missing this common CFTR deletion indel when you're looking at your patient's data. Another thing that makes it more complicated is we think of the reference genome as a thing that exists. It's a true thing. It's a reference genome. It's actually not any one person. It's a bunch of people put together that we've decided it's the reference, but at least we all agree that this is what the reference is. This is what we're comparing against. And yet people are still working on the reference, and so it's getting better over time. But that means it's changing over time, which is also not intuitive. And so we, for a lot of our data, have been using the HG37 reference build, but we're starting to move to HG38 so that when you're looking at the same variant, again, this CFTR variant here, you're seeing that the reference, the address of the variant is totally different depending on what reference build you're looking at. And that's mostly because the non-coding portion of the genome is being filled in with additional detail. So it's just important that you're looking at your data in the right reference build. We actually loaded a project with the wrong reference build and spent a few hours trying to figure out why the analysis wasn't working before we fixed that. Variant quality, there's a lot of metrics we use for variant quality. I'm just going to cover a few of them here. So this is what our data kind of looks like. So this is our family ID here. This is a SNP, so it's a single nucleotide variant. So an A to a G is just a heterozygous site. And then you have this 99 down here. That's the genotype quality. 99 is the best score you can get. It caps at 99. That means it's a high quality variant. I just think of higher being better. The next factor is the depth, which is just the number of reads that cover the site, which you see here, 54. The next is the allele balance, which is the number of alternative reads, how many reads cover your variant versus how many map to the reference, so that you can get a sense. So in general, you would think it'd be, for a heterozygous variant, it'd be about 50% of your reads capture the variant, about 50% capture the reference. We see a little bit of a skew against the against the variant, but in general, it should be around close to 50%. This one's 31%, seems reasonable-ish, a little bit low. If it's very low, you worry more about a sequencing artifact or a somatic mutation. And then there's something called VQSR that GATK, the genome analysis toolkit that we use to call variants at the Broad, uses. And it's called a variant quality score recalibration, but it's really just another way of giving a quality score to a variant. And for the most part, it's a machine learning approach, and it just says, does this variant that I'm asking about look more like a good variant or more like a bad variant? And it kind of puts it in one of those bins. It's a complicated topic. You can read all about it if you want to get more into it, but that's mostly how we use it. So things that pass VQSR are better variants, or more reliable variants. Okay, transcripts. Transcripts make things much more complicated. So your variant annotation may differ completely depending on what transcript you're talking about. And then the community has two main different transcripts sets that we use, RefSeq, which is from the American transcripts from NCBI, and then the GenCode annotations through Ensemble from European Institute. And so research labs tend to use a little more Ensemble. For a lot of what we do, we use Ensemble transcripts, but a lot of the clinical labs tend to use RefSeq, so there is importance of knowing which transcripts you're talking about, and there are programs that will help you go back and forth between the two. We talk a lot about the canonical transcript. It tends to be the longest transcript that includes all of the exons. There are a few exceptions to that, but that's what I typically mean by that. And then, of course, for whatever disorder you're thinking about, there might be one transcript or a few transcripts that are more clinically relevant than other transcripts. And that's also something that people are working on curating and sharing more of that information. So this is just showing you here. This is the RefSeq transcripts in blue on the top for MECP2, which is going to be one of my favorite genes for this presentation. And then the Ensemble transcripts on the bottom. There tends to be more Ensemble transcripts for most things I look at. And then you can see that just there's some shorter versions, some longer versions. There's a lot of variety. And on what I have down here is on the Nomad browser, which I'll be introducing more later, where you can see the gene here at the top, which way it's going, and then some of the different transcripts and some expression data here, which we'll talk more about. Okay, so variant nomenclature. We basically want to describe the variants using a C-dot coding nomenclature of the nucleotides and a P-dot amino acid nomenclature. And we use and follow the rules of the Human Genome Variation Society, or HGVS. I'm just going to kind of briefly introduce a few of these. But basically, HGVS annotates versus the transcript. So if it's a reverse strand transcript like I was showing you, it annotates versus that, whereas the VCF uses the plus strand or the 5' strand and annotates everything versus that. So you'll see some discrepancies there. The synonymous variants or silent variants are places where the nucleotide sequence changes, but the amino acid doesn't change, as opposed to non-synonymous or missense where the amino acid changes. The in-frame indels, I think of them a lot like missense variants. They tend to delete one amino acid or a few amino acids. So this is an example here of a 3' base pair deletion that results in a 1 amino acid deletion. And then we have our protein truncating variants, or a putative protein truncating variants. So these are nonsense variants or stop-loss variants, frameshift variants, and splice site variants. So one of the concepts we use is called worst annotation. So while something might have one effect on maybe the canonical transcript or the clinically relevant transcript, we always give a variant the kind of worst annotation it can get. And so I'm going to show you some examples of these. So these are different exons or in the different transcripts for a gene. The actual specific gene doesn't matter. And you'll see that there's this one exon here that's only in one of the many transcripts. And so if you happen to have a patient that has a nonsense variant in that exon, that's going to come up in your VCF as a nonsense variant. It could even be a known disease gene. You could think it's really interesting, but then when you look, you say, well, it's only in an exon that's in this one transcript. Is that even important? And so it would actually be called as an intronic variant in all these other transcripts. And so you have to really take that into account. And we see these coming up all the time actually in the Nomad data set, and that these variants are in the general population because they are not really deleterious. So you have to think about that, but we're going to call it a nonsense variant, even though in many contexts it's an intronic variant. Here's another example down here. This is, to me, just sort of funky looking. Mostly this is a very large last exon, but there's this one transcript that has a smaller last exon and then this like tiny little exon. And so there are a bunch of splice variants, essential splice variants being called around there. And so when you look, you can say, well, that's a little bit odd. I'm not really sure that it would really splice like that or why it would do that. It's only in one of the transcripts. And then you can also use expression data on top of that. And so this is showing mRNA expression level. And you can see that sort of these two transcripts are more highly expressed and this transcript isn't expressed. So the fact that there are splice acceptor and splice donor variants around this unusual exon was a way that I could figure out that these weren't biologically important. Okay, so then we can think about using the power of allele frequency, which Daniel has already introduced. Because if we just look at all of the variants in an exome, there are thousands of them, and so really by figuring out which ones are where, we know where to focus our attention. There are a larger number, a growing number of reference population databases that are available these days, and I'm going to talk just about a few of the ones that we use most heavily, but they're all listed there, and you can check out other ones if you're interested. So 1,000 Genomes is one of the first reference databases that was around. It involves sequencing exomes and low coverage genomes from very diverse ancestries, and because of the ancestral diversities, we still use it today. EXAC was the first aggregated exome reference database developed at the Broad Institute in the MacArthur Lab, and it has broad representation from 5 different ancestries. Rather than sequencing all the data directly for this project, it was actually aggregated from data that had been sequenced for a number of common disease studies. And when released in October 2014, really changed the molecular diagnostic field as it became the control database for molecular interpretation. Bravo is the largest, currently today, Bravo is the largest genome reference database. It's through the TopMed project. There are some restrictions on data sharing, so they cannot share ancestry and you can't download the whole data set, but you can, they have a website that looks very much like EXAC that you can look variants up, and a lot of the data is in dvSNP that you can check it out there also. One caveat is they do have a few most likely have related individuals in the data set, so you have to be a little bit careful when thinking about rare variants. I like to mention this other database, the Greater Middle Eastern database. It's pretty small and it includes individuals with Mendelian disease or relatives of individuals with Mendelian disease. But Middle East is one of the areas that we don't have good representation in any of the available reference databases, and so sometimes it's helpful to go and look up a variant here, particularly if you're analyzing a patient of Middle Eastern ancestry, because you can find that it's actually a population variant that's just not well represented in our current databases. And then most of what I'm going to talk about is going to focus on GNOMAD. So GNOMAD is the Genome Aggregation Database. It has data provided by 107 different PIs from many studies. Over 138,000 individuals are included, so there's 123,000 exomes and over 15,000 whole genomes. It's all Illumina data. It's all processed through the same pipeline, and it's all called as one giant data set. And this is really what the power of the data is, that normally data is processed in different ways, but here all of this data has been very uniformly processed and all jointly called. The entire site's VCF for each position is downloadable. The individual level data is not. And you can annotate any of your data set with the allele frequencies from this data, from the GNOMAD. We don't have phenotype information for the individuals in the data set. Hopefully, as new studies happen and we build biorepositories, more of this will happen in the future, but this is not what GNOMAD was set up to do. We include people who have disease because everyone in the general population eventually has some disease, so there's not a completely healthy population out there. But what we do is we take studies that have cases and controls from common disease studies, and we don't knowingly include anyone from a Mendelian disease cohort, particularly a pediatric onset Mendelian disease cohort. We have one new population in GNOMAD versus EXAC, which is the Ashkenazi Jewish samples, which have been pulled out, and the male-female balance is pretty even. It's about 55% male, and the mean age is 54 years. So most people who have these variants have lived a reasonably long time with them. And then we report the highest population allele frequency, which we call the PopMax for each variant, and I'll talk more about that later. So this is GENOMES. This is the principal component analysis. And what this does is how we determine ancestry. So you can see in orange over here are the Europeans, and the Finnish and Ashkenazi Jewish are the populations that are right next to it. See, these are weird colors. This here, I think, is the East Asian. Down here is the Latino. Up on the top is the South Asian, and over here is the African. And so by taking individuals with known ancestries and seeing where they fall on these maps, we were able to then classify the rest of the individuals in GENOMES with a lot of confidence. We do remove samples from GENOMES that have low quality. We also remove individuals with sex chromosome abnormalities, and we remove first and second degree relatives, which is really important so that you can know that when you see a variant in two individuals, they're not at least a parent or child or a second degree relative. So we have a browser, the GENOMES beta browser. There's actually two versions of this. So one is at gnomed.broadinstitute.org, and one is the GENOMES beta one, which is most of my slides will come from, and I really recommend people check out. So the browser was developed by Conrad and Ben, and now is innovated and maintained by Matt and Nick. So you can look up any gene there. So I typed in MECP2. This is a smaller gene. It's one of the reasons I like showing it. So you can see it has 3 exons. In blue is the coverage of the exons. And you can see it's about 90x in some regions. It dips in some areas. These are probably like higher GC content areas. And then in green is the genome coverage. And you can see that's pretty even across the gene. It's a little dip there. I'm not sure why. And then below that, you have all of the variants that are in the gene. I've actually clicked missense and loss of function here, so I'm not showing you the non-coding variants, but you could click all and see those. You can also change which data set you're looking at. If you only wanted to look at EXAC, for example, or only the genomes or exomes, you can change this here. The default is that the whole VCF is available through the browser, but we only show variants that pass the filter. But if you had some variant that you were really interested in that you see in a patient, you might want to check if it is failing the filter, because there are real variants that are back there. They're just enriched for artifacts. And then a really nice feature, we have this search bar, and you can search for various things here. So one of the things you can do is you can look up a specific variant that you're interested in. You can use the HGVS to search, or the chromosome coordinate, or you could even search on the term missense if you wanted to look at every missense variant. So I wanted to look for a specific threonine variant here, and I actually just typed it in. I think this is neat. I'm not totally sure what context I would use it in, but it does highlight all the threonine variants in this gene. I always think that's a fun thing to do if you were looking for any kind of pattern there. But I decided I wanted to check out this threonine 240 serine variant. So I clicked on that link, and it takes me to the variant page. There's a page for every single variant in the GENOMES data set. At the top, it tells you the total number of the allele count for exomes and for genomes, and the total number of people genotyped at these sites in these data sets, and the allele frequencies in exomes and genomes. There's a link to dbSNP, a link out to UCSC, and a link to search if the variant is in ClinVar. There's some metrics at the top. I don't use those that much. You can click on the transcripts here, and you can see where the variant falls in various transcripts. So you can see it's a missense variant in both, but the numbering changes depending on which exons are included in the transcript. And then we use polyphen and SIFT there if you wanted to see some in silico analysis. So this is where the data I'm interested in. This is the population allele frequencies. So the allele count is the total number of times that variant has been called. So homozygotes would have an allele count of 2, and heterozygotes would have an allele count of 1, and it just adds them all up to get the total allele count. The allele number is the number of chromosomes genotyped at that site. So it's that number divided by 2 is the number of individuals. This is a variant on the X chromosome. So we can actually, when we count the number of homozygotes, these are all female by definition. For autosomes, you'll just see homozygotes. You won't see hemizygotes also. So there's 2 homozygotes for this variant. And then we have the number of hemizygotes, so there's 44 total, or 38 in the most prevalent population. So the next thing I wanted to talk about is the idea of PopMax. So you have your allele frequency for each ancestry group that we have included in the data set. You can see the global down here. It's highest in the Africans here at 0.8%. It's much lower at some of the other ancestries, including not being seen in the South Asians at all. So it's just interesting. You can see the difference for carrier frequencies across different populations. And then I want to introduce the concept of filtering allele frequency. So basically, we tended to use these allele frequencies because these are large populations. So 17,000 chromosomes. This is 8 to 9,000 individuals who are genotyped at this site. But basically, all of these are a sampling of the general population, and so we can correct these statistically to develop confidence intervals to say how confident are we in this sampling of the population. We're going to be more confident when we've genotyped more people and less confident when we've genotyped fewer people. And so we can calculate the 95% confidence interval of this max number, and so we're calling this the filtering allele frequency. And I'm going to get to that a little bit more later of how we use this, but just wanted to point out that for EXAC, it's on every page, and we're working on including it for NOMAD. So the next thing I do when I look up a variant in NOMAD is I check out the read data supporting the variant, and this is a really crucial step. So this is that threonine missense variant to become serine, and you can, so a single nucleotide variant. Up here you have the sequence. This is just showing you it's in exons, it's in all three transcripts for this gene. Then this is the reads, this is the coverage up here, so you can see it's about half blue for this variant and about half orange for the reference, and so that looks like a pretty, 50-50, looks pretty good. And then you can scroll down here, which you obviously can't do on my slides, but you can see that the variant is falling in multiple reads. If you scroll down, it's in a lot more reads. There's a little bit of a more pink reads up here. These are different strands of the reads, but when you scroll down, it looks really good. And there's no other variants in the area. This is the homozygote here and a hemizygote, and those also look very clean. So most of the variants we look at do look like this. Not super exciting, just reassuring that this variant looks fine. So what are you looking for when you're looking at the IGV read visualization to see if you have any questions about the reads? So one of the things is we've flagged variants that are low confidence loss of function variants. That's not to say that the variant isn't real, it's just to say that just because a variant is called a protein truncating or loss of function variant, it may not actually result in protein truncation. And so it's important to look at the variant and see what you think would happen from it. We have poorly aligned regions, multinucleotide variants, and somatic mosaicism, and I'm going to go through each of these. So this is what the low confidence loss of function variants. We use LOFTY, which was developed by Konrad Karczewski, to look at loss of function variants and determine if there's any concern about it causing loss of function. So an example is that nonsense variant in an exon that wasn't included in any of those transcripts. Often those exons are not well conserved, and so we find those and call those dubious transcript annotations. We're not sure those are real loss of function. And then the other thing is protein truncating variants that occur near the end of the gene in the last exon or in the 50 base pairs of the penultimate exon shouldn't trigger a nonsense-mediated decay, and in many cases, those won't result in loss of function. There's times where the last exon, you lose some repressor domain, or the last exon is very big, and so truncating it will still cause other problems, but it at least shouldn't cause nonsense-mediated decay. And so we have flagged those with this low confidence loss of function tag. Okay, poorly aligned regions. So sometimes you just see a region that looks like this. This kind of raises a lot of red flags for me that something weird is going on here, and I don't really trust any of the variants in the area. You can see that these two variants are in cis for the most part, but there's some scattered variants that are in only one read. These ones have different allele balances, that they're very low allele balances compared to these over here. We have an insertion over here. There's just a lot going on. And what's happening actually is if you look at the sequence at the top, you can see it's very blue. There's a lot of C's up there. And this is just a low complexity repeat. And these are areas that are probably hard to sequence through, but mostly they're very hard to map through because of the low complexity. So we tend to see more sequence artifacts there, and so we have flagged these as low complexity repeats so you can be aware of them, or low complexity regions. Homopolymers. So these you can also just see in the read data that you have about 8 Gs there in a row. These are areas that tend to accumulate PCR artifacts during the exome sequencing. However, there are also regions where the human polymerase has trouble, that these homopolymer runs are tricky, and so you can have real mutations arise there. So overall, you can't rule it out completely. Whether if you're just seeing it, it's hard to say. But these are just areas I would definitely think about Sanger sequencing before believing it's real. Multinucleotide variants, these are some of my favorites actually, just because the VCF is actually interpreting every variant separately, so these aren't getting picked up the same way that we think about them. So what's happening here is you have two variants within the same codon in cis, and each one is being called as a variant 1 or variant 2, and so one's a missense and one's a nonsense, but they're in cis, they're in the same codon, they're one thing, they're not two variants. If they were in trans, they'd be two variants. And so really what's happening is a 2 base pair substitution, and so it's a serine to glutamine missense variant, so it's an entirely different missense variant than you were first thinking about. And so it's just important to be aware of these. They're super easy to spot if you just look at the read data, because there's two colors right next to each other, and so that would flag your attention. We flagged these in EXAC, and we're working on flagging them in NOMAD. The same kind of thing can happen for frameshift events or complex indels. So you could have, for example, a 2 base pair deletion, go down a few bases, and then have a 2 base pair insertion that restores the frame and only changes the amino acids that are sort of in between those two events. And so that's another time where you might get all these frame shifts called, but when you actually look, it's just one complex rearrangement in the region. We see these happening where you might have 2 or 3 and they're in just one person, and they're getting called as a bunch of different frame shifts. So that you can also see on the read visualization. And skewed allele balance. So what's happening here is you have, this is a C in the reference, but it's a T variant in this person here. And when you look, it's actually, the T is only in 16 of the reads, so only 21% of the reads, which raises some chance that it could be mosaic. 21% is high enough that it could just be that your sampling is a little bit off and it's really a heterozygous variant. But it does sort of raise your suspicion about it. So one of the things is that we have flagged, we have filtered these, in EXAC, they're just there. But in NOMAD, we've actually taken allele balance into the quality metrics and the quality control we've been doing on NOMAD. So more of these are getting filtered if there's a systematic bias in the allele balance. Which is what that does. Okay. So often the variants I'm most interested in are those that are absent from NOMAD, because if they're not in the general population, maybe there's something interesting going on. That being said, we all have variants that aren't in NOMAD, and we're all basically doing okay. So that's not the end-all be-all, but still those are the variants I'm going to zoom in on and think about more carefully. One of the things to be really careful about, though, is when I say a variant's not a NOMAD, why is it not a NOMAD? There can be a few different reasons. One is that whatever variant I'm thinking about, I'm actually using some kind of description of it that doesn't match what we're using in NOMAD, and so it's really there, but I just don't see that it's there. And so it's very important to think about using chromosome coordinates, so we're all talking about the same variants. The second is that maybe the position isn't covered in NOMAD. It's a little bit better with the genomes now that we often have at least coverage in 15,000 people, but particularly for EXAC, and there's still some regions of the genome, there are just places where we may not have good coverage due to mapping problems or other issues, and so it's very possible that your variant isn't there because we don't have any data there. And third, the variant is really not in NOMAD. We have lots of people genotyped at that site, and we can say with confidence it's not there. So there's various ways to look up and see if a variant is in NOMAD, but this is sort of the quickest way to do it. Pick your chromosome coordinates. Don't worry about if it's a C to a T. Don't just pick the position and search for that position. Because what that's going to do is it's going to give you a 50 base pair window around the position, and it's going to show you all the variants there. I like to click including the filtered because it gives me all the variants then, because what I'm really interested in is not so much is my variant there, because I think my variant isn't there, but what is the coverage of the other variants in this region. So first you can see this region doesn't have great coverage. So the average coverage is only about 20 down to 10x in the exomes. That's not great. The genomes are pretty smooth at 25, 30x. And so when I look at look for my variant of interest, I've picked that I really want to find this proline 273 threonine missense variant. I don't see that in the data set, but I do find a synonymous variant at that position that's been filtered out. And so I can look there. I can Okay, I can look there, and I can look at the allele number, so I can see the number of chromosomes that have been genotyped at that site, so 65,000 chromosomes, so about 32,000 people have been genotyped at this site. And so while I can't say the variant is absent from all of NOMAD, implying 138,000 people, I can say it's absent in 32,000 people, which is still a pretty good thing to, reasonable thing to say. And so just that you can use NOMAD to figure that out. Okay, so that's mostly what I'm going to cover for reference population databases, but I wanted to show you how we're using the data. Sorry, I feel like a lot of my stuff is in this corner here. So we annotate the whole, so this is what a line of our VCF looks like, although I've kind of rearranged it to fit on a slide. You'll see it later in this afternoon. So this is the proband who's affected because they're shaded in, and they're male because they have a square. This is the father because he has a square, and this is the mother. This is showing that the variant is present in the proband, and it's a deletion of three bases. The father is wild type, and the mother also has this variant. With the chromosome coordinates over here, you can link out to UCSC. It tells you what kind of variant it is, and it also tells you the HTTPS PDOT nomenclature. So for every single variant, we have annotated it in our VCF with the allele frequency in 1,000 genomes, in EXAC, and in NOMAD. And so we can see what the population max allele frequency is, and we can also see the number of homozygotes. And when we hover over, it tells us how many people, what the allele count is for this allele frequency. Down here there's 5. The allele count is 5 here, and that this is the number of people genotyped at that site. So we have all that information that we can annotate for our VCF with. There's a bunch of other things we can look at, though. So in silico predictions, there's many in silico predictions. It's a huge goal of the field that we can develop a computer algorithm that can pinpoint which variants are disease causing and that we should be focusing on. And so there's a lot of work that's gone into this. They're useful tools, but they overall don't kind of hit their mark yet, and that we're not really diagnosing cases, at least in our hands, based on these. But they use a combination of conservation, amino acid characteristics, and protein structure and other features to infer pathogenicity. So there's some that are really designed for missense variants and only give a score for missense variants. There's some that work for all variants, like CAD and IGAN. And then one of the things to really be aware of with these is you can't just add them up and say, oh, well, I have 2 red and a yellow, which these are just based on our kind of cutoffs that we use internally. And we have to use our own internal cutoffs for this color scoring because they all use different kinds of numbers, and so it's really hard to remember what scale goes with what. But they're very circular in that they're all using a lot of the same information. So even if they all agree, it can be that they all agree because they're basing their determination on similar features. And most of them are trained on the same data sets because we only have so much truth data. So they tend to be a little bit circular in how you use them, and you just have to keep that in mind. So we look at them and we think about them because they're easy to generate, but we don't want to overinterpret them. Even though many of these scores use conservation, I tend to look at conservation also. So this is a serine, sorry. This was actually this variant here, so this is the deletion of this serine at position 372. And so I think about this sort of like a missense variant. Is this very conserved? If I deleted it, would I expect it to cause a problem? And you can see it's pretty conserved. It's a C or cysteine in some places. Okay, so another approach we use is frequency filtering. So this is kind of getting back to how we use our reference population databases. This is work by Eric Minikel, James Ware, and Nikki Whiffen. So I've told you about NOMAD and EXAC and how having these large population databases is really powerful. And part of that is because when we looked at what we had before, the exome sequencing project, which was a database that was an order of magnitude smaller and also only had Europeans and African Americans, if we used a 0.1% allele frequency and just said how many variants are so rare that they only occur 1 in 1,000 people, using this as a reference population database, we still had close to 600 to close to 1,000 variants that we had to look at that were considered very rare by this metric. And you can see that they worked better for ancestries that were this African and European on the bottom here because that's what the control population was, and they worked less well, and you had more variants to look at for populations that weren't well represented. And so with EXAC, we went from 6,000 people to 60,000 people, so an order of magnitude in increase in size of the control population database. And so the number of variants we had to review that were very rare fell by about 5-fold, which is great. It's still 150 to 200 variants. That's still a lot to look through. So by that metric, the rarer you go, the fewer variants you have to look at. And so you want to be able to push your allele frequency thresholds down as low as you can so you have fewer variants to look at, and yet you don't want to filter out any of the variants you're interested in. So the question becomes, how common is too common for a variant to be pathogenic? So our standard approach is for recessive variants to use a 1% allele frequency filter. There are some founder variants that have a higher allele frequencies, and so you may probably want to check for some of the known ClinVar things or HDMD and make sure you're not missing anything. But for the most part, this is how we sort of standardly do our recessive searches. For dominant, the standard has been 0.1%, but this leaves a lot of variants to review, as I showed you on the last slide. And for de novo dominant, that's a lot. That's a pretty high allele frequency filter to be using for a lot of conditions. And so frequency filtering is based on the idea that a pathogenic variant in a reference sample can't exceed the prevalence of that condition in the general population, taking into account in the calculations penetrance, though. So if a condition that was highly penetrant has a prevalence of 1 in 10,000 in the general population, then I wouldn't expect to see a variant at an allele frequency higher than 1 in 10,000. And in fact, because we think we have depleted NOMAD for people with Mendelian pediatric onset conditions, I would actually think the variant might be even lower than that. So for any disease you're thinking about, you can actually calculate your disease-specific allele frequency threshold pretty easily by thinking about the genetic architecture of the condition. So you just take the disease prevalence times the genetic heterogeneity and divide by the penetrance, and that gives you your maximum credible population allele frequency. Nobody really wants to do the math, so we have a calculator that was developed by James Ware and Nikki Whiffen that you can do these calculations. And what's nice about that is then you don't even have to say, I'm sure that these are the numbers I want to work with. You can slide the numbers around and you can see how it would work with different models. So this works pretty well for dominant disease. For biallelic recessive disease, honestly, the numbers tend to be pretty big still, and so you're welcome to try it. It's definitely good to explore, but I mostly use this for dominant disease. So their disease of interest is hypertrophic cardiomyopathy. It's generally a dominant disease, so we mark monoallelic. It has a prevalence of 1 in 500 people in the general population. Allelic heterogeneity is 3%. Seems like a totally random number. The way they get this is they just say, what is the most common variant? If you take all the people with hypertrophic cardiomyopathy, what variant has the highest prevalence in this disease population? And that actually is a specific variant in MYBPC3. Sorry, I normally have this up on the slide. I don't have it on this one. And so the allele frequency of that in their really large hypertrophic cardiomyopathy cohort is like 2.3%, and so they take the confidence interval of that, and it was about 3%, so we use 3% as the most common you would expect a single variant to be contributing to this disease. But you can do different things with this number depending on what your disease areas of interest is. And then penetrance is tricky, but they're starting with a penetrance of 50% in hypertrophic cardiomyopathy. It's not well estimated for a lot of disorders. And basically that when you put all these numbers in, you get a maximum credible population allele frequency up there, and it's 6 times 10 to the minus 5, which is really low. It's much lower than this 0.1% I've been talking about, or 1 times 10 to the minus 3. And so you can start to use these allele frequency filters levels to do your filtering, and you'll have many fewer variants to look at. And then you want to compare this not necessarily to the global allele frequency, although you could, but actually the most stringent is to compare it to the filtering allele frequency, which is our confidence interval adjusted pop max allele frequency that's been adjusted for the sample size. So these again are available in EXAC, and we're hoping to have them on NOMAD soon. Okay, I'm going to switch topics to talking about constraint. Probably most people have heard about this, and this is other data that has come out of the reference population data sets and has shown how we can really find new disease genes and new biology by studying not what we see in the general population, but what we don't see. So this is work by Caitlin Samoha, who is a graduate student with Mark Daly, and now is a postdoc at Sanger, and also Konrad Karczewski, who's working on it for Nomad. Okay, so the idea is mutations are going to arise basically at random based on kind of sequence context across the genome. And so it's not that certain genes are going to have a lot of mutations versus others. They should all kind of acquire mutations at a similar rate. So as these mutations arise in different individuals in the population, we're not actually seeing historically what's happened. We're seeing which mutations have been passed on to people in the present day. And so for a tolerant gene, most of those mutations will get passed on. Most of those variants will get passed on just by chance. A few won't. A few might be deleterious, and a few just won't get passed on. In a constrained gene, mutations are going to be deleterious to the function, and so these aren't going to get passed on because of there'll be negative selection against them. And so for these, what we're seeing is what's happening today in the general population. Tolerant genes will have a lot of variation because it'll arise and be passed on, and constrained genes will have little variation because it will arise, but it won't be passed on. So Caitlin developed a complex model where she looked at the sequence context, which took into account the length of the gene, and then she corrected it for coverage in the data set, and also divergence between humans and macaques, so if there's regional divergence in the data. And she was able to, for each gene, say how many variants of each type do we expect to see in today's population. So this is a known disease gene, JAG1. It causes Alagille syndrome, which is a dominant haploinsufficient congenital disorder affecting the liver, heart, and eyes. It has pretty high penetrance but variable expressivity, but many children are pretty severely affected. So what we find is that in this gene, we expect to see about 235 synonymous variants, and she sees 200, which is about what's expected. For missense variant, she expects 478. She sees a little less than 300, which is actually a divergence with a z-score of 4 from what she expected. And for loss of function variants, and importantly, she's only counting essential splice site variants and nonsense variants because it's hard to develop a mutational expectation for frameshift variants. But looking at these two variant types, she expects to see 45. She only sees 1. And so this has a very high PLI score of 1. I can tell you what PLI is, though. So PLI is the probability of loss of function intolerance, and it's estimating whether a gene is thought to be haploinsufficient. And haploinsufficiency is just when, in a diploid organism, having only one functional copy of the gene isn't enough for the wild-type phenotype. So you could be haploinsufficient, could be a characteristic, or it could be a disease, but it's just the phenotype is different than the wild-type phenotype. We tend to think of high PLI genes as causing disease since there's constraint and selection against them in the population. So again, PLI, probability of being loss function intolerant, and we call a PLI greater than 0.9 to be sort of abnormal. These are the genes that we're interested in. We think these are our candidate haploinsufficient genes. We don't use z-scores because a little bit differently than the missense, because the expectation is so much lower, like an order of magnitude lower. There was some correlation between the number of loss of function expected and the length of the gene. And so she had to develop another algorithm to sort of take that into account. So really importantly, haploinsufficiency, PLI, dominant disease is what we're thinking about. Same for a missense constraint, dominant disease genes. So recessive disease genes, we have carriers in the general population, so we see variation in these genes in the general population. It might be a little bit less than you would for a non-recessive disease gene, but overall there's enough variation that they're not depleted to the same extent, and so these scores don't necessarily and generally don't pick out recessive disease genes. The other thing is if you have a disorder like BRCA1, where the phenotype is breast and ovarian cancer, very severe disorder, but this is happening mostly after the age of fertility. Many people are passing on these what are viewed as deleterious variants before, to their offspring, and then developing the phenotype later. And so you're not seeing the same evolutionary signature for those types of later onset conditions. So high PLI, missense constraint, really good for pulling out disease genes that cause more of a pediatric onset phenotype. So there's 3,230 genes with a high PLI score from EXAC. Those numbers are going to change a little bit from Nomad. And when Caitlin first released these, over 70% had not been assigned a phenotype in OMIM. I actually feel like that number has gone down a lot because we're all focusing so much on these genes. The second is missense constraint. So there's about 1,800 genes that are missense constrained across the whole gene. we call missense constraint having a z-score greater than 3. Caitlin then went and tried to find regions of the gene that were missense constrained. And so that work is still on bioRxiv, and I'm going to tell you a little bit more how you can access the data more easily. And then the final is the MPC score, which is a site-specific score for missense variants. I mention it because you'll see in the list of in silico analysis that we're doing, it's just another one of these scores, and it takes into account how depleted a specific change is of one amino acid to another in the general population, constraint, and polyphen. Okay, so all the constraint data is available on the browser. You can find it in 2 places. One, it's on the EXAC browser, and 2, it's on the new Nomad Beta browser. When you go to the Nomad Beta browser, there's a table on top for each gene. It, by default, shows the Nomad data, and so the constraint scores are still only released for the EXAC data, and so you can click on EXAC here, and you can see this table. We're working on getting them for the Nomad data, so hopefully we'll have those sometime this summer is the goal. But the other thing is the other thing that's coming soon is regional missense constraint to be on the EXAC site. So this is what it's going to look like here. This is CDK13, which has 2 regions of regional missense constraint. And so it shows you the amount of depletion. So this only has 28% of expected, and this has 7% of expected missense variation. Okay. There are a large number of other reference data sets that we use from the literature and other reference databases. Some of these I'm just going to mention. So obviously PubMed. When we find variants that we think are interesting, we read about the genes in PubMed. We use gene cards to read about what the genes do, the Protein Atlas for expression, and the GTEx portal. Monarch is you'll be hearing more about later in this course, has cross-gene and phenotype resource across different species. Decipher I'm going to show you, and UniProt is another protein resource. So I'm going to very briefly introduce some of these. So this is OMIM, Online Mendelian Inheritance in Man. For all the known disease genes for Mendelian disorders, there's a large text description that's a curation of the literature, and then you can click on this clinical synopsis and you can get all the clinical features of the condition. So that's very helpful when you find a variant in a gene you think looks interesting. When you are looking at the phenotype of the person whose data you're analyzing, you can go to OMIM and you can look at the phenotype there and you can compare those. If I have a variant in a gene where I know what the gene does or don't, one of the things I might think about is what tissues is this gene expressed in, because that could impact what phenotypes it might be likely to cause. So I'm showing MECP2 here, which is associated with Rett syndrome, which is affects females and is a neurodevelopmental regression disorder. But this is just to show an example. So what you're seeing here is the expression across 53 different tissues from individuals who donated their bodies to science, and different tissue biopsies were taken at autopsy, and then RNA sequencing was done across them. This is the largest data set I'm aware of that shows expression data, and it also does splicing analysis on some of this data. So it's a really great data set to have. There's a GTEx portal, so all this data can be easily accessed, and you can type in any gene. And then what you see here is I've put these in the order, so you can sort them alphabetically, or you can sort them in order of expression. There's some other features through other menus at the top that you can do. And so you can just see, so brain is in yellow here, and you can see it's highly expressed in certain regions of the brain, but it's pretty well expressed across all the tissues I look at. The other time I really use this data set is if I wanted to do a functional study. I would want to know what tissue I need to get to do said functional study. Daniel told you the example of just because the gene is expressed doesn't mean the splicing pattern or other expression will be the same as what was happening in your disease. But just in general, I wouldn't want to do a skin biopsy to look at expression level and then find out that it's not even expressed in skin. I had wasted my time when I could just check out this data set. So MECP2, again, if I wanted to do some kind of assay, I wanted to look if I can get a blood sample. This is whole blood here. This is the TPM, which is the transcripts per kilobase million. So it's 12, which is pretty good, from 407 samples in the adult population. I will say, like children, the data might be, children or fetal, the expression might be a little different, but this still gives you really useful information. In general, if the TPM is at least 1, you can quantify whether the gene is expressed or not. And if the TPM is at least 4, you can try to get some evidence of whether the variant is impacting splicing. So it's just very helpful to know where you should be looking. I don't tend to use protein data as much, but a lot of people really like it. So this is the Human Protein Atlas for MECP2, which shows you the same thing as the RNA expression, that it's just expressed in a lot of different tissues. This is the time where it's more helpful. I might say I have this variant in a gene that I think the phenotype is muscle disease, and so could this variant in this gene of interest be causing muscle disease? And so I want to look and see where the protein or the RNA is expressed, and oh look, it's expressed in heart muscle and skeletal muscle. That seems like a good candidate. If I was instead interested in hearing loss, and I wanted to know whether this gene could be causing hearing loss, and this was the expression profile, I might say, well, that doesn't really look like it fits as well. So this information can be very helpful and is very easily accessible. You'll be hearing lots more about these, but these are basically databases for variant interpretation where something is already known or in the literature about that specific variant. So ClinVar from NCBI is an open access data set of clinical interpretations, mainly from clinical labs, but also from OMIM that has been added from the literature. One of the tricky things is much of it comes after ACMG criteria, which you'll hear more about, that really is the standard for the field so that we're all talking about the same thing when we're talking about variant interpretation. But some of it does come from before, and that's confusing because the standards before were often that if a rare variant was seen in a patient with disease, sometimes it was called pathogenic without having anything else. But as we know from GENOMES, we all have lots of genetic variation, and that most of it is not causing disease. The Human Gene Mutation Database is a pay access database that is curation of the literature, but again has the same kind of issues that many things are in the database that actually are not disease causing. And then the Leiden Open Variant databases are other databases. They're normally gene or phenotype centric that researchers and other people are putting these data sets together. They're great resources for the community, but again, not everyone's not using the same standards, and so that can be confusing. This is from Decipher. It's one of my favorite ways to look up a gene. And so I just want to highlight a few things here and encourage everyone to use it. So you can check it out on Decipher at Sanger. It's the protein view of the gene. It has a ton of information in this one view. So you have the exons of the gene. This is our friend MECP2 that we've been talking about all morning. This is the burden of nomad missense variants. So you can see there's a lot over here, not as many over here. Nomad hemizygous variants, because this is an excellent gene on the X chromosome. And then nomad loss of function, which are clustering here, which makes me think that there's something going on over here. I guess it's in the last exon, so it's not causing loss of function there. That makes sense. You can have your protein domains here. Your data up here is the variants from Decipher, depending on your phenotype. So this is neurodevelopmental intellectual disability, so I would expect it to be in some of the data sets that are included in Decipher, like the Develop DDD project. And then this is all the ClinVar data down here. So you're seeing a lot of triangles. What's going on with those? In red are putative protein truncating variants. In yellow are missense variants. The filled triangles mean that someone has said it is pathogenic or likely pathogenic in ClinVar, if it's below the line. And the open ones is either, I think it can be benign or likely benign, but they're mostly VUSs that are included on this data, on this view. So there's tons of VUSs in this gene. And then you can see some pathogenic, likely pathogenic missense, and then lots of protein truncating pathogenics. The regional constraint data is currently available on this view. So this is another gene, KMT2C, which is associated with Kleefstra syndrome. And you can see that this is the regional missense constraint. There's a constrained region here, constrained region here, and a constrained region here. And then these are like just minorly depleted, and then the scale is over here. So if you needed to access this data now before it's on the GENOMES site, this is where you can come to find it. And then just to briefly touch, there are many variants that are predicted to affect splicing. The ones we really think about are the ones we assume that these essential splice sites, the +1, +2, -1, -2 sites would affect splicing. For everything else, there are certain regions that are suspicious. They're somewhat marked in this table. Based on the consensus sequence and what we know of disease mutations. But honestly, this is just complicated, and we often use something called Alamut, which is another, you have to pay to access it, a resource where you can put in any variant. This is showing, it runs 4 different splice detectors here. This is for a splice splice acceptor site. It's saying the splice site would be here in the reference, and when we put in our variant of interest here, it's shifting the splice site by 2, and so this would result in a 2 base pair out of frame insertion. So it's just another tool, so something to think about beyond coding changes or things that can affect splicing. So this can happen with synonymous variants near the ends of exons. It can happen with intronic variants that are either deep intronic or shallowly intronic. So there's other things to think about. Okay, and so this is putting it all together. This is the same variant I showed you earlier. This variant is actually in ClinVar as pathogenic, and so it gets annotated as such. It's an indel, so CAD scores come up for it, but none of the missense tools will actually report a score on this. We have our reference population data. So we take this all together, and this is the type of data we think about when we annotate our VCF, when we try to find the variants that are causing the disease of interest. So in conclusion, really all talking about the same thing, having correct variant annotations and the information that goes along with it is really crucial. Reference population databases have been really critical in our ability to identify rare variants, which is an important step to figuring out which variants could be causing disease. That being said, most variants are rare and don't result in disease, so it's only one necessary but not sufficient criteria. And then we take all of these, we apply filters, and then we search deeply in the literature to try to diagnose patients with rare disease. And so this workshop that we'll be doing shortly after this will give you a chance to take all of these features and dive in to try to analyze cases, including some undiagnosed ones. So we'd love everyone's input if they're able to diagnose any of those, because these are all patients out there that are still searching for answers. And so then just to thank everyone, including Daniel and Heidi, and then Sam, who's been and Danielle have been organizing this course. And Ben and Hannah are the software engineers that write the Seeker tool that you'll be trying out this afternoon. And I stole some slides from Matt, so thank you. I can take some questions.